How did humans discover medicines and know they treat specific diseases? When you think about how advanced modern medicine is today, it's incredible to realize it all started with ancient observations, natural remedies, and a lot of trial and error. But how exactly did we get from chewing on leaves to designing drugs with artificial intelligence? Let's break down the fascinating journey of how humans discovered medicines and figured out which ones work for specific diseases. The beginnings: trial, error, and observation. In ancient times, people didn't have microscopes or clinical trials. They had nature, intuition, and observation. Early humans noticed that chewing certain plants reduced pain or fever. For example, the willow tree was used to relieve pain and fever. It contains salicin, a compound related to modern aspirin. Ancient Egyptian, Chinese, Indian, and Greek civilizations all had herbal medicine traditions. At this stage, medicine was about what seemed to work, not why understanding why it worked. From herbs to active ingredients. As time passed and science developed, people began to ask, what's the actual substance in this plant that makes me feel better? This led to the discovery of active compounds, the specific chemicals inside plants or minerals that have biological effects. Examples: quinine from the bark of the cinchona tree was used to treat malaria. Morphine from the opium poppy became a powerful painkiller. Scientists began to extract, purify, and synthesize these substances, laying the foundation for modern pharmacology. Scientific method and clinical testing. Eventually, medicine moved beyond trial and error. It became scientific. Here's how modern drug discovery works today. Step-by-step drug testing. Lab research, in vitro. Test on cells in a petri dish. Animal testing, in vivo. See how the substance behaves in a living organism. Clinical trials, on humans. Multiple phases to ensure safety and effectiveness. Phase 1, is it safe? Phase 2, does it work? Phase 3, how well does it work and what are the side effects? And Phase 4, long-term monitoring after approval. This process can take 10 to 15 years and cost billions of dollars. Understanding the disease equals targeted treatment. One of the biggest breakthroughs in medicine came when we started understanding diseases on a deeper level. Instead of just treating symptoms, doctors and scientists began to ask, what's causing the disease at a cellular or molecular level? Bacteria, virus, genetic mutation, immune malfunction. This led to antibiotics for bacterial infections, for example, penicillin, antivirals for viruses, like HIV treatment, and targeted therapies for cancer and autoimmune diseases. The better we understood the cause, the better we could design specific drugs to counter it. The modern age, AI, big data, and personalized medicine. Today, drug discovery is more high-tech than ever. AI is used to predict which molecules might work as drugs. Computer-aided drug design helps build molecules virtually before testing them in labs. Personalized medicine allows treatments to be tailored to your DNA. We're moving from one-size-fits-all to precision medicine, which is especially important in cancer treatment, neurology, and rare diseases. So, how do we know a drug treats a specific disease? It's all about scientific validation. A hypothesis is formed. This drug might work. It's tested extensively in labs, animals, and people. Data is collected and analyzed. If it consistently works and passes safety standards, it becomes an approved treatment. Regulatory agencies like the FDA in the US, EMA in Europe, or MHRA in the UK oversee the process to protect public safety.